Hi everyone, wish you and your family to be in good health during the outbreak of this deadly coronavirus. I welcome you all to the class 3 of enzymology, and today we'll be studying about enzyme inhibition. So why enzymes need to be inhibited? So first of all, it is a cellular control mechanism to regulate different metabolic processes. For example, the enzymes which are allosteric in nature, so they can be controlled by addition of small groups or ions which act as inhibitor. So addition of these groups alters the structure of the enzyme. So now they are conformationally changed so that the substrate can no longer bind to the enzyme, and this reduces the enzyme activity. And when the cells again need to reactivate the enzyme, they remove these groups so that the enzyme is again activated and the metabolic process will be faster. So this is a very important control mechanism. We'll be studying about this control mechanism later on in enzymology. It is a mechanism through which drugs, toxins and poisons act. So this is an important mechanism used by scientists and druggists to synthesize novel molecules which exhibit their toxicity by adding or by modifying the enzymes which are essential for survival of the organism, so that they will be specifically killing those organisms but not harming the other organisms. So these inhibitions also provide important information about the transition states through which the substrate passes when it is converted to product. So recall from the previous classes that the substrate is converted to product by passing through a transition state, and many molecules which are analogous to this transition state can be used as inhibitor to study the structure of this the transition states. Lastly, inhibitors provide very important information about the active site residues and the mechanism of their action. So the active site amino acids can be identified by addition of different amino acid inhibitors, so that the enzymes are no more active. For example, serine. Serine is present in chymotrypsin in the active site, and this serine can be inhibited by addition of a serine inhibitor, so that now when the inhibitor the serine, the OH group, which is a nucleophile in chymotrypsin, is permanently modified so that it doesn't activate the process. So these are some of the important informations that the inhibitors provide about the enzymes. So in the subsequent videos, we will be studying about different types of inhibitions and what happens to the enzyme kinetics during these inhibitory processes.